Hi, myself Nirmala Swami. I have completed MSc in Biotechnology. Currently, I am doing PhD in Bioinformatics. In this lecture, I have talked about molecular docking. Mainly, we are going to discuss about what is molecular docking and their importance, types of molecular docking, uh, advantages and disadvantages, some of the important software used for docking in these sections we have provided. Now we are going to see what is molecular docking. Before we should know what is docking. Docking is to find the best matching between two molecules, whereas molecular docking is to predict the structure of intermolecular complexes between two molecules. For example, protein-protein, protein ligand, protein nucleotide. It is optimize the conformation and the relative orientation between protein ligand. We are going to see importance of docking. It is extreme relevance in cellular biology as well as molecular biology. It is used to identify the novel drug designing or discovery of drug design discovery, and also it is used to repurposing the drug, which means existed drugs we can recreate it, and also it is used in CAD, computer aided drug design. Here we are going to see types of docking. There are two types of docking. One is rigid docking, another one is flexible docking. Rigid docking means it's also called lock and key. In this docking method, protein and ligand, the structure will be rigid, which means unbending or unfolding or unbending and flexible structure will be there. So this whole also is one of the drawback of this software, which means the ligand structure is conformational won't change. So your dock with the protein and ligand means it will go and bind. It's not flexible, so it won't bind proper way. So it is the one of the drawback. Whereas flexible docking, it's also called induced fitting. It is generate the conformational libraries. It's also called flexibos. It contains 10 to 20 conformations for each ligand. It depends upon the software, docking software we are using. So some of the software will contain more than 20, 50 confirmation changes also. So what is the advantages of using docking? It provides the confirmation with a favorable surface. Whereas the disadvantage is it does not provide the high degree of accuracy, and also in the binding modes. Whereas the limitations of docking, it provides the less accuracy, and also it ought to predict the binding energy, also the function score. This is the flowchart of the work doing docking. Target selection. Target selection, we are using protein selections. The uh, protein software house Uniprot, and also protein data bank. It's called PDB. And the ligand selection, we are using PubChem or drug bank. Once the structure was downloaded from the respective softwares or databases, we will have to prepare the certain targets or ligands for the particular software, docking software. What they requires depends upon the software. We have to prepare the target and ligand. Then prepared molecules we should dock to each other, and we can evaluate the docking results. So here I am provided the protein selection databases Uniprot and PDB. Uniprot is the one of the software which used for protein, whereas the PDB is a 3D dimensional structure it will give. So Uniprot and PDP is interlinked databases. Here I am given Uniprot. Uniprot in this option, if we are giving the protein name, it will give that two categories. One is Swissprot and another one is TRMPL. Swissprot is the manually annotated and TRMPL is computationally annotated. So in this blue color highlighted, it shows the entry. Entry means the Uniprot KB ID here provided, and this is the entry name which gives the protein name along with the organisms. So protein name it's in short form, here it's in abbreviational form. Here it's respective to reviewed one or unreviewed one, means Swissprot or TRMPL, here they mentioned. Here the gene name and here the organisms. It's the if you are providing the protein name, here means it that protein is belongs to which family you want, I mean which organisms you want, here they will provided all those things, and the sequence length also they will provided here. So if you want more details about protein also, if you are clicking this entry and you can go into that Uniprot page. And this is PDP protein data bank. If you know the protein name, you can search in option, you can provide the protein name, or else you can provide the 3-4 letter code. So in the head they will mention all those information about the protein structure and how what are the methods they are used, the protein whether the protein contain the mutation or structure it's talked with some other protein or ligand information also they will provide in here, and also they will attach a literature report also, so we can download the paper and we will read, we will get to know the protein informations. So ligand selection means there are two software and databases I have provided. One is PubChem, another one is DrugBank. This is the URL link. If you need to go means you just click on, you can go to the link. The PubChem, this is the PubChem database. I have given for example one drug name or compound name. You can also give your area of, I mean your compound interest, and you will get all those informations about the compound. Other software also is there, another software that is called drug bank. Here also they will provide the summary and about the about the drug informations they have provided. So docking software, in this section I am given I have categorized three software list. One is online, another one is installed, another one is purchased. The three different categories docking software I have provided along with the URL link. If you need, you can go to into the link. If you need a detailed information about these softwares and the importance why we are using this particular each server, all those information I have given my next video. Just follow my channel. So SwissDoc, ZDoc, HDoc, Doctor, PatchDoc, these are online server. You can click the link and you can go to the page. And these are the installed software: Autodoc, Pyrex, iGemDoc, Doc. And Scodinger is purchased software. If you have fun for purchasing software means you can directly go into Scodinger. So these are the two papers I have referred for this lecture. If you need more information about software and with along with the information means you can directly go to the directly follow the paper, and some of the informations I have get it from online server databases, websites, articles and books. I am sincerely thank all of them, and almost I have thanked my Vellore Institute of Technology. Thank you so much.